Okay, good afternoon. Thank you for the chance to speak here. Today I'll be talking about how machine learning can help us reduce the noise and improve the quality of the data that we get from Histone ChIP-seq experiments. So this is joint work with my collaborators Emma Pearson and Anshu from Stanford. Okay, so as you all know, Histone ChIP-seq measures the presence of histone marks that lie along the genome. So here's some sample data from a lymphoblastoid cell line. Right? The x-axis is genomic location. Each different bar represents a different histone mark, and the y-axis is represents the degree to which that mark is present at that location in the cell population. And I'm preaching to the choir here, so of course we are interested in this because it can give us some insight into the regulation of our genome. The problem, though, is that ChIP-seq can be expensive and finicky. It often requires a lot of sequencing and a lot of input material. So in practice, the data that we get can be quite noisy and hard to work with. So this example we show here looks quite nice, and it was actually sequenced to quite a high read depth, more than 100 million reads per mark. So it looks good, but it's also very expensive to do. On the other hand, if we just sort of subsample this and just took 1 million reads per mark, you can see a significant sort of degradation in quality. It's much noisier. It's quite hard to work with this data and analyze it properly. On the other hand, it's also just much easier to run this experiment and much cheaper. So in an ideal world, we would run this sort of cheap and easy experiment, but still get the data that was as good as what we saw on the previous slide. Okay. So that might sound impossible, but there's some reason to believe that we can do something about it. And indeed, in other domains, people have considered and tackled this problem. So let's consider images. Okay, so we have a clean image, and we add some noise to it. Okay, it becomes grainy, but so long as you don't add too much noise and completely overwhelm it, then we can tell what the original image was. And if my Photoshop skills are better, I might even be able to recreate the clean image from the noisy one. In fact, others have shown that you can train machine learning models to do this, to take in a noisy image and try to reconstruct what they think the clean image was. This works even in more challenging settings, such as image inpainting, when instead of adding random noise, we just occlude a whole part of the image. Even though part of it is missing, as humans, we can make a good guess as to what lies behind that occlusion. And likewise, people have shown that machine learning models can also make such a guess. And most of the time, they do pretty well. So this is for images, but the same has been done for many other data modalities like noisy speech and even text. So some of you might have seen this. Right? Most of you can probably read it. Even though there is some noise in the data, the letters are scrambled, and we still can understand what it means. And the takeaway is that if the data is highly structured, right? If not all the pixels in your image are completely sort of independent, or if there's some correlation between letters in a word, then even if you have noisy or partial observations of the data, you can still try to get closer to what the true underlying data is. Okay, so that's going to be the idea that we are exploring in this talk. And after all, we know that biological data is indeed highly structured. So here's the plan. Right? We're going to generate or take some existing high-quality data, maybe expensively obtained. And then we're going to fix a given type of noise. So as an example, let's consider the noise introduced when you have a very low sequencing depth. Okay? So once you fix that type of noise, we can generate or obtain a matching noisy version of the high-quality data. So in our example, we can take the reads from this high-quality experiment and just subsample it, which gives us noisy data. And once we have this matching pair of noisy data, we can train a model that learns a mapping from the noisy data to the clean data. And finally, once we have this model, we can then apply it to other data that we haven't seen from, say, different individuals, different cell lines, that for which we might only have the noisy data but not the high-quality one. Okay, so this is the general idea. In particular, we use convolutional neural nets as our model. So for each output mark, we train a separate CNN, one for the signal, so fold change prediction, another for peak calling. And each of these networks uses all of the input marks, all of the histone marks as input, but it just outputs a single one. Okay, so we train a sort of aggregation of them. And we use CNNs because the convolution operation lets us keep the model quite compact and concise. And also we use a lot of computation as we scan across the genome, which lets us have a more expressive model at little computational cost. But I do want to stress that while CNNs we think are a good fit for this data type, it's nothing about the magic of neural networks or anything that actually makes this work. It's just we want some sort of statistical model that can learn to transform noisy data into clean data. Okay, so how well does this work? What we did was train this model on the lymphoblastoid cell line data derived from one individual of Central European ancestry. And then we tested it on a cell line from a different individual, right, this time of Chinese ancestry. Okay? So the model only sees one cell line during training time. Here we see the test input. So at the top, this is the noisy data that we fed into the model. This is sort of a very downsampled version of the cell line derived from the Chinese individual. And at the bottom, this is the desired output. It's what we hope the model would produce. And indeed, the model's output actually looks quite similar to the target output, at least quite different from the noisy input. Right? And using more quantitative metrics, like mean squared error and correlation, the predicted output of a model using just 1 million reads per mark was actually equivalent in quality to what we'll get if we use 15 to 25 million reads per mark instead and just did sort of a standard processing. And so this can be a sort of significant improvement in the quality of your data. And we found that this pattern held up across all the histone marks that we looked at for both signal prediction, which is on the left, and for peak calling, which is on the right. And each individual dot on this plot is a different test individual from different ancestry. Moreover, we confirmed that the model was not just copying the training data, the high quality training data, over to the test data. To do this, we looked at the regions that were significantly different between the training set and the test set. Right? And we found that even on those regions, which the model hadn't sort of seen before, we were able to obtain quite significant improvements in the quality of the signals and peak calling. And moreover, the model is doing something more than just simple linear smoothing. So we compared it on these regions to a linear model, so like linear regression or logistic regression, and found that the convolutional neural net also did quite a bit better. Okay, so all in all, we applied this to 3 different sources of noise. We've seen the first one from low sequencing depth. We also tried it on cases where the noise comes from low amounts of, sort of input cells as well as low enrichment antibodies. And so in general, we found that it worked. So for the low cell count case, instead of computationally adding noise, which is what we did when subsampling for the sequencing depth case, here we just use experiments run on different amounts of input data, right? Input material. So for the noisy data, we use experiments run on 100 cells, and for the high quality data, we use experiments run on 10,000 cells. And then we, as usual, train a model to learn the mapping between these. Right? We found that even in this more challenging setting, the denoising model was able to help. And this, we think, could be especially useful, especially, say, when working with primary data, primary tissue, where input material is often limited. And for the third example we showed, we simulated the effect of using an antibody, very low specificity or sensitivity. So if you have sort of a low enrichment experiment, then what happens? So we simulated this by taking a high quality experiment and then throwing away 90% of the reads and replacing it with reads from the control experiment, which doesn't use any antibody. Right? So this basically just washes out most of the signal. But nevertheless, we were able to get most of it back using the models. And moreover, when we did sort of other tests, like looking at aggregate enrichment plots at DNase 1 hypersensitivity regions, we found that the denoising models did something that was kind of reasonable and closer to the true data. Okay, so let me wrap up. We've presented an approach to sort of denoising that relies on being able to generate matching pairs of noisy and clean data, and then learning the mapping between them. Okay? And we think that this is applicable, so not just to histone ChIP-seq, but also just to situations in which these criteria are fulfilled, right? Where you have a lot of structure in your data, where it might be hard to model the noise analytically, which is sort of true in almost all biological applications, but it's sort of comparatively easy to generate pairs of noisy and clean data. It might take a lot of money at first, but once you have that model, then you can apply it as much as you want. Okay. The main caveat, of course, is that we rely on one big assumption, which is that the noise processed in the training data is the same as that in the test data. Okay, so you can't use this as sort of a magical thing to just remove all the noise in your data if you don't have a corresponding training set, which for different types of noise, we might not know how to generate. It's also not always easy to tell if you just have noisy data. How good is the denoise output? Is it still noisy? Is it now super accurate? How much can you trust it? So that, I think, is an interesting direction for future work. How do you just make this more robust? And of course, you can extend it by using other data sources, such as DNA sequence or sort of reference epigenomes, like in the Ernst and Kellis imputation work. And all of these should help to improve the power of the denoising models. In general, we are optimistic that these computational approaches will just become an increasingly important complement to improve experimental techniques in terms of getting us data that is high quality and yet sort of easy to do at larger scale and not so expensive. Okay, so thank you. This is, we call our model CODA, of the convolutional denoising algorithm, and our code is all up on the CODA repository, including scripts to download the data and reproduce the results that I've just seen. This, once again, is joint work with Emma and Ansho, and thanks a lot for listening.
Hi, nice work. So I was wondering, normally you have like training and validation of your model, but it looks in this case that the validation is quite correlated to your training because you have the same sort of high quality output that you expect. Or am I mistaken? Can you train your model on the noise of the experiment of less reads and then validate it on a different genome? Or
yeah, that's a good question. So in general, because our assumption is that we want the noise in the training set to be the same as the test set. So the closer you can get that, the more effective it's going to be. So if you do it in one individual and then you test in a different individual, that's going to be more effective than if you do it in one species and test on a different species, right? So in our paper, we have run experiments on sort of transferring the model across individuals, across different cell types, and then across species. And it gets worse and worse as you go on, but still does reasonably well. Yeah. And the validation to pick model parameters and hyperparameters is done in a standard way. So we hold out part of the genome. So we hold out a couple of chromosomes, and we use that as validation to pick our hyperparameters. And that's still all in the training set. And then when we test it on the test set, that's unseen. Yeah.
Good work. I was wondering about whether your tool would also work for TF ChIP-seq, because here you're using Histone. Have you also looked at TF, where the signal is is sharper?
Yeah, great question. We haven't tried it, but I think it will not work as well without augmenting it with some of these other data sources. Right? And the reason is that because it's so sharp, there's less structure. Like if you miss the peak, you're not going to see a peak next door that will tell you. Whereas here, if you have a broad peak and you're missing a chunk here, but there's another mark that's correlated that you do observe, then you can sort of fill that in. Yeah.
Hi, maybe I missed a little detail. So are you looking at each track separately, or do you subsample the whole, let's say, epigenome that you get with several different histone modifications, as you always showed on your plots?
So each model, we use jointly the set of all histone marks that are available to us in order to make our predictions. But in this particular model set that we have chosen, we train models to output individual marks separately.
Okay, but I mean, if you have only limited input, let's say only one or two or three of the tracks, would it still work? Or?
Yeah, so it actually works quite well if you just use the mark that you're trying to output. So you have noisy data for, say, K27 acetylation, and you just use that to output high-quality version. That still works pretty well, but the more input data you add, then the better it does. Yeah.
Have you tried to interpret the model to see which tracks are like predictive of which other tracks?
Mm-hmm. Yeah, great question. So only a little bit, and more as a sanity check. You observe like correlations between and anti-correlations between the different histone marks as you might expect. But we've only seen those in large trends like this. We look at the filters learned by a neural net and like it's high in one mark and low in another mark. But we haven't tried to see if we can tease out anything sort of more biologically meaningful. I think it would be interesting, especially to use this in conjunction with like sequence data, to try to learn sort of motifs where, you know, maybe given a particular motif, this mark is correlated with that, but with another motif, it's not, and that could be interesting.
And one more thing, the window size is 25 KB, I think.
Yeah.
Is it something also you optimized using the hyperparameter selection? Like the full window
so
length?
let's see. Yes, so this was, we tried a bunch of different lengths of input into the model and just picked the one with the best validation accuracy. I don't think there's anything super special about this set of hyperparameters, and in general, if you're trying it on a different set of noise, the different type of noise, different data type, it's quite likely that something different would be better. Mm-hmm.
All right, let's thank Wei once again.
Great, thank you.